Dear friends, good morning. Myself Dr. Chandrasekhar Bangarwal, Professor Department of Kriya Sharir, SRM State Ayurvedic College, Bareilly, Uttar Pradesh. We were talking about the resistance and immunity. We have seen that there are two types of resistance: non-specific resistance, which is also called as innate defense. Another is specific resistance, which is also known as immunity. We have also seen that there are two types of immunity: cell mediated immunity and antibody mediated immunity. Today, we will learn about the antibody mediated immunity, hypersensitivity, and types of acquired immunity. This part of your syllabus we will cover today. Now, antibody mediated immunity. The body contains not only millions of different T cells but also millions of the B cells, each capable of responding to specific antigen. Cytotoxic T cells leave lymphatic tissues and seek out and destroy a foreign antigen, but B cells stay put. So there is slight difference between the functioning of T cell and B cell. Similarity is that there are millions of the B cell and T cells in the body. Now, in the presence of antigen, specific B cells in lymph node, spleen, or mucosa associated lymphatic tissue becomes activated. They then differentiate into plasma cells that secrete specific antibodies which circulate in the lymph and blood to reach the site of invasion or where exactly the infection takes place. Now, during activation of B cells, an antigen binds to the B cell receptor. Antigen is produced by the viruses or bacteria. These integral transmembrane proteins are chemically similar to the antibodies that eventually are secreted by plasma cells, which are formed by the B cells. Although B cells can respond to an unprocessed antigen present in lymph or interstitial fluid. Their response is much more intense when they process the antigen. That means antigen processing is also necessary. Now, activation of proliferation and differentiation of B cells. Antigen processing in B cell occurs in following ways. First of all, antigen produced by the viruses or bacteria is taken into B cells. Then these antigens broken down into peptide fragments and they combine with MSC2 self-antigen and move to the B cell plasma membrane. Now here, helper T cell recognize the antigen MSC2 complex and deliver costimulation required for the B cell proliferation and differentiation. Now, the helper T cell produces interleukin 2 and other cytokines that function as a costimulator to activate B cells. Interleukin 4 and interleukin 6 are produced by helper T cell, enhances B cell proliferation, B cell differentiation to plasma cells and secretion of antibodies by plasma cells. Now some of the activated B cell enlarge, divide and differentiate into clone of antibodies secreting plasma cells. After few days after exposure to antigen, a plasma cell secretes 100 millions of the antibodies each day for about 4 to 5 days until plasma cells dies. Now, most antibodies in the lymph and blood to invasion site. They travel to the site of invasion Some activated B cells do not differentiate into plasma cells but remains as a memory B cells that are ready to respond more rapidly and forcefully if should the same antigen reappear at the future time. Now, different antigens stimulate different B cells to develop into plasma cells and their accompanying memory cells. All of the B cells of particular clone are capable of secreting only one type of antibody, which is identical to the antigen receptor displayed by B cells that first responded. Now, each specific antigen activates only those B cells that are predestined to secrete antibody specific to that antigen. Antibodies produced by the clone of plasma cells enter the circulation and form antigen-antibody complexes with the antigen that initiated their production. Antibodies and antibodies can combine specifically with epitope on the antigen that trigger its production. The antibody structure matches its antigen much as lack accept of a specific key. Plasma cells could secrete as many as different antibodies as there are different B cell receptors because the same recombined gene segment code for both the B cell receptors and antibodies eventually secreted by plasma cells. Now antibody structure. Antibodies belongs to the group of glycoprotein called globulins. So they are also called as immunoglobulins. Antibody action. How antibody act? Antibody can act in the one or many of the following ways. Number 1, neutralizing antigens. Number 2, immobilizing bacteria. Number 3, agglutinating and precipitating antigens. Number 4, activating complement. And number 5, enhancing phagocytosis. These are the 5 ways how the antibodies can act. Number 1 is neutralizing antigen. The reaction of antibody with antigen blocks or neutralizes some bacterial toxins and prevent attachment of the some viruses to body cells. Immobilizing bacteria. If antibodies form against antigen on the cilia or flagella of the motile bacteria, the antigen-antibody reaction may cause bacteria to lose their motility, which limit their spread into nearby tissues. Third is agglutinating and precipitating antigen. Because antibodies have 2 or more sites for binding to antigen, the antigen-antibody reaction may cross-link pathogen to one another, causing agglutination, clumping together. Phagocytic cells ingest agglutination by microbes more readily. Likewise, soluble antigen may occur out of the solution and form more easily phagocytized precipitate when cross-linked by antibodies. Number 4 is activating complement. Antigen-antibody complexes initiate the classical pathway of the complement system. We will see later on how complement system works. Now number 5 is enhancing phagocytosis. The stem region of an antibody acts as a flag that attract phagocytes once antigens have bound to the antibody's variable regions. Antibodies enhance the activity of phagocytes by causing agglutination and precipitation. It activating complement and by coating microbes so that they are more susceptible to phagocytosis. Now we should remember important part. There are 5 types of the antibodies. Number 1 IgG, number 2 IgA, number 3 IgM, number 4 IgD and number 5 IgE. We should not forget that Antibodies are also called as immunoglobulins, and if you remember, this is a part of the blood. They are also found in the blood plasma. These antibodies are created in the liver. Now, first antibody, IgG, is the most abundant. About 80% of all antibodies in the blood found in lymph, blood, or intestines. These IgG protect against viruses, bacteria by enhancing phagocytosis, neutralizing toxins, triggering complement system. It is only antibody to cross placenta from mother to fetus, conferring considerable immune protection in newborn. This is the specialty of the IgG antibody, which is 80% of the total immunoglobulins. Second one is type A, which is found only in sweat, tears, saliva, mucus, breast milk, and gastrointestinal tract secretions. Smaller quantities are found in blood and lymph, makes up about 10 to 15% of the antibodies in the blood, occurs as monomers or dimers. Monomers means a single unit and dimers is a two units together. When during stress, the level of this Ig antibody increases, lowering resistance to the infection. This provides localized protection on mucous membrane against bacteria and viruses. Now, third immunoglobulin is IgM, which is about 5 to 10% of the antibodies in the blood. This IgM also found in lymph. It occurs as a pentamers, that means 5 unit. First antibody class to be secreted by plasma cell after an initial exposure to any antigen. Active complement system cause agglutination and lysis of the microbes. Also present as monomer on the surface of B cells where they serve as antigen receptors. In blood plasma, anti-A and anti-B of ABO blood groups which bind to the antigen A and B during incompatible blood transfusion are also IgM antibodies. Nowadays, whenever a person go for the test for corona positive or corona negative, these two types of antibodies are focused. One is IgG and another is IgM. If IgM level is increased, then it is concluded that that patient has recent infection and he should be quarantined. If IgG levels are increased, then it is concluded that he could have been infected by the coronavirus but need not have quarantine because IgG indicates a past infection and IgM indicates a newer infection, first exposure to any antigen. Now IgD, it mainly found on the surface of B cell as antigen receptor where it occurs as monomer. IgD involves in the activation of B cells. It makes about 0.2% of antibodies in the blood. Last type of immunoglobulin is IgE, which is less than 0.1% of all antibodies in the blood, occurs as monomers. This IgE located on the mast cells and basophils. IgE involved in allergic and hypersensitivity reactions. IgE provide protection against the parasitic worms. This is the summary of the type of antibodies or type of immunoglobulins. Again, remember that these immunoglobulins are produced by the plasma cells and B cells are converted into plasma cells. These antibodies or immunoglobulins also found in the blood plasma and they are produced in the liver. Now, let us see about the role of complement system in immunity. The complement system is a defensive system made up of over 30 proteins produced by the liver and found circulating in blood plasma and within tissues throughout the body. Collectively, the complement protein destroy microbes by causing phagocytosis, cytolysis, and inflammation. They also prevent excessive damage to body tissues. Most complement proteins are designated by an uppercase letter C, numbered C1 to C9, name for the order in which they were discovered. The C1 to C9 complement proteins are inactive and only become activated when split by enzymes into active fragments, which are indicated by lowercase letter A and B. Now, for example, inactive complement protein C3 is split into activated fragments C3A and C3B. Active fragments carry out destructive action of C1 to C9 complement proteins. Other complement proteins are referred to as the factor B, D, P. In all properly in P means complement protein act as a cascade. One reaction triggers another reaction, which in turn triggers another reaction, and so on. With each succeeding reaction, more and more product is formed so that the net effect is amplified many times. Complement activation may begin by three different pathways, all of which activate C3. One activated C3 begins cascade of reaction that brings about the phagocytosis, cytolysis, and inflammation. The reaction takes place as follows. Number 1. Inactivated C3 split into activated C3A and C3B. Number 2. C3B bind to the surface of microbes and receptors of all phagocytes attached to the C3B. Thus, C3B enhances phagocytosis by coating a microbe, a process called as opsonization. Opsonization promotes attachment of the phagocyte to the microbe. Now C3B also initiates series of reactions that brings about cytolysis. First, C3B splits C5. C5B fragment then bind to the C6 and C7, which attach to the plasma membrane of an invading microbe. Then C8 and several C9 molecules join the other complement protein and together form cylinder-shaped membrane attack complex, which insert into the plasma membrane. Now number 4. Membrane attack complement complex creates channels in the plasma membrane that result into cytolysis, the bursting of the microbial cell due to inflow of the extracellular fluid through the channels. C3A and C5U binds to the mast cells and cause them to release histamine that increase blood vessel permeability during inflammation. C3-5U also attract phagocyte to the site of inflammation. Now C3 can be activated in three ways. One is classical pathway, starts when the antibodies bind to the antigen. The antigen-antibody complex bind and activate C1. Eventually C3 is activated and C3 fragments initiate phagocytosis, cytolysis and inflammation. Alternative pathway does not involve antibodies. It is initiated by an interaction between lipid carbohydrate complex on the surface of microbes and complement proteins factor B, D and P. This reaction activates C3. Third is lectin pathway. In the lectin pathway, macrophages that digest microbes release chemicals that cause liver to produce protein called lectin. Lectin binds to the carbohydrates on the surface of microbes, ultimately causing activation of C3. Once complement system is activated, protein in the blood and on body cells such as the blood cells break down into activated C3. In this way, its destructive capability is very quickly so that damage to the body cell is minimized. Now, let us summarize the cells participate in the immune response. You can remember antigen presenting cells. There are three: macrophages, dendritic cell and B cells. Lymphocytes, they cytotoxic T cells, helper T cells, memory T cells, B cells and plasma cells and memory B cells. Let us see one by one. Macrophages, phagocytosis, processing and presenting antigen, foreign antigen to T cells. Secretion of interleukin 1, which stimulates interleukin 2 by helper T cell and induce proliferation of B cells. Secretion of interferon that stimulate T cells growth. What is the function of dendritic cell? Processing and presenting antigen to T cell and B cells found in mucous membrane and lymph node and skin. What is function of B cell? Processing and presenting antigen to helper T cells. Now lymphocytes. Number 1 is cytotoxic T cells. Kills host target cells by releasing granzymes that induce apoptosis. Perforin that forms channels to cause cytolysis. Granulocyte that destroys microscope. Lymphotoxin that destroys target cells DNA. Gamma interferon that attract macrophages and increase their phagocytic activity. And macrophage migration inhibition factor that prevent macrophage migration from site of infection. This is speciality of the cytotoxic T cell. Then helper T cell cooperate with B cell to amplify antibody production by plasma cells and secrete interleukin 2, which stimulate proliferation of T cells and B cells, may secrete gamma interferon and tumor necrosis factor, which stimulate inflammatory response. Third one is memory T cell, remains in the lymphatic tissue and recognize original invading antigen even years after the first encounter. Now B cells differentiate into antibody producing plasma cells. Now plasma cells, descendant of B cells that produces and secrete antibodies. Memory B cells, descendant of the B cells that remain after an immune response and is ready to respond rapidly and forcefully should the same antigen enter the body in future. Now let us see about the types of immunity. There are 4 types of immunity: naturally active immunity, naturally passive immunity, artificial active immunity and artificial passive immunity. That means type of immunity depends on whether it is natural or artificial, whether it is active or passive. Let us see one by one. Regarding naturally active immunity, following exposure to microbes, antigen recognition by B cells and T cell and co-stimulation lead to the antibody secreting plasma cell, cytotoxic T cells and B and T memory cells. This is generally occurs during infection. Now naturally passive immunity transfer IgG antibodies from mother to the fetus across placenta or IgG antibody from mother to the baby in milk during breastfeeding. Here the passive means baby has nothing to do. He is ready-made getting some IgG antibodies from mother through placenta IgG antibody and during breastfeeding IgA antibody. This is passive process, but this is natural. That's why it is called as naturally passive immunity. Now artificially active immunity. Here we are doing something artificial. How actively we goes? Antigen introduced during the vaccination stimulate the cell-mediated and antibody-mediated immune responses, leading to the production of memory cells. The antigens are pre-treated to be immunogenic but not pathogenic. That is, they will trigger immune response but not cause significant illness. This happens during all type of vaccinations like BCG, MMR, or poliovirus and all. Now artificially passive immunity, when you by injection intravenous injections of immunoglobulins are given to the patient. This is example of artificially, patient has to do nothing, he is ready-made getting antibodies. Let us come to the next point, hypersensitivity. A person who is rare overly reactive to a substance that is tolerated by most other people is said to be allergic or hypersensitive. The antigen that induce an allergic reactions are called allergens. That means immune system does not work only during the infection. Overall, this immune system works to fight against the infection, bacterial, viral, fight against the toxins, fight against the different types of tumors include cancer, and at the same time this immune system also works during the allergy. During allergy, the antigen that induce an allergic reactions are called allergens. Whenever an allergic reaction takes place, some tissue injury occurs. Now, from where these allergens can enter the body or from where it gets, common allergen include certain foods like meat, peanuts, shellfish, eggs, antibiotics like penicillin, tetracycline, vaccines like pertussis, typhoid, venoms like honeybee, wasp, snake, cosmetics, chemicals in the plant such as poison ivy, pollens, dust, molds, iodine containing dyes used in a certain X-ray procedure, and even microbes. These are the examples from where the allergens are produced in the body when they come in contact with the person who is hypersensitive. Now there are four types of hypersensitivity reactions. Type 1 also known as anaphylactic, type 2 known as cytotoxic, type 3 known as immune complex, and type 4 known as cell mediated. Out of these four, first three are antibody mediated immune responses and fourth one is cell mediated immune response. Type 1 anaphylactic. Anaphylactic reactions are the most common and occurs within few minutes after a person is sensitized to an allergen is re-exposed to it. That means in first time patient does not have anything, second time it has reaction. In response to certain allergens, some people produce IgE antibodies that bind to the surface of mast cells and basophils. Next time same allergen enters the body, it attaches to IgE antibodies already present. In response, the mast cells and basophils secrete histamine, prostaglandin, leukotrienes, and kinin. Remember that this happens second time, not the first time. So first time patient does not experience anything, but the second time a reaction occurs which is known as anaphylactic reaction. Collectively, these mediators cause vasodilatation, increase blood capillary permeability, increase smooth muscle contraction in airway of the lung, and increase mucus secretion. These four serious things occur. As a result, a person may experience inflammatory response, difficulty in breathing through constricted airway, and runny nose from the excess mucus secretion. In anaphylactic shock, which may occur in a susceptible individual who has just received a triggering drug or being stung by the wasp, wheezing and shortness of the breath, as airway constrictions are usually accompanied by shock due to vasodilatation and fluid loss from the blood. This life-threatening emergency is usually treated by injecting adrenaline to the dilate airways and strengthen the heartbeat. This is common emergency occurs in the clinics of a doctors. Secondly, cytotoxic. Cytotoxic reactions are called antibodies IgG or IgM directed against antigen on the person's blood cells like RBC, lymphocytes or platelets. The reaction of antigen-antibody usually lead to the activation of complement. Type 2 reactions which may occur in incompatible blood transfusion reaction damage cells by causing lysis. So during blood transfusion this care is taken. There should not be cytotoxic reaction. Immune complex, third type. Immune complex involves antigen, antibodies and complement, IgA and IgM. When certain ratios of antigen to antibody occurs, the immune complexes are small enough to escape phagocytosis, but they become trapped in the basement membrane under endothelium of the blood vessel, where they activate complement and cause inflammation. Glomerulonephritis, rheumatoid arthritis occurs in this way. Fourth is cell-mediated. Cell-mediated reaction or delayed hypersensitivity reaction usually occurs 12 to 17 hours after exposure to an allergen. This reaction occurs when allergens are taken up by the antigen-presenting cells, such as the Langerhans cells in the skin, that migrate to lymph nodes and present the allergen to T cell, which then proliferate. Number 4 is cell-mediated. Some of the new T cells return to the site of allergen entry into the body, where they produce gamma interferons, which activate macrophages and tumor necrosis factor, which stimulate inflammatory response. Intracellular bacteria such as Mycobacterium tuberculosis trigger in this type of cell-mediated immune response, as do certain haptens such as the poisons or IV toxins. The skin test for tuberculosis is also delayed hypersensitivity reaction. So this is about the hypersensitivity. I am sure you will be able to understand all the things. For this purpose, you have to listen this video or video related with immunity more than one time, at least 3-4 times. When you listen carefully and read the matter, you will be able to understand. In the next lecture, we'll just sum up immunity and a new experimentation we are going to do. We are going to change the language. In Hindi language, we will sum up all the immunity reactions. Thank you very much. Have a good day.